The overall goal of this procedure is to simultaneously map transmembrane potentials and calcium transients in HL-1 atrial monolayers loaded with both calcium and voltage dyes. This is accomplished by first culturing the HL-1 cells on a cover slip to a proper confluent density. The second step is to load the calcium and voltage-sensitive dyes into the cells. Next, the HL-1 monolayer is stimulated with a bipolar electrode. The final step is to record the action potential and the calcium transient signals by fluorescence microscopy. Ultimately, the action potential and calcium transient traces can be compiled to show propagation of the signals across the confluent monolayer over time.
This method can help answer key questions in the cardiac electrophysiology field, such as how calcium transients and action potentials are related in the electrophysiological properties of cultured HL-1 cells subjected to various drug treatments.
Though this method can provide insight into patterns of action potential and calcium transient propagation at microscopic level, it can also be applied to biological systems such as pathologically remodeled human hearts and arrhythmogenic animal models.
Generally, individuals new to this method will struggle because HL-1 cells maintaining cardiac myocyte phenotypes are difficult to culture, and a carefully optimized protocol is needed to culture, pathogen, and stain the monolayer.
To begin, make all of the stock solutions for the experiment as indicated in the text protocol. Grow the HL-1 cells in T25 flasks and replace the medium with 5 ml.